Hello, everyone. Thank you for inviting. It's really a great pleasure to be here with you today, and I'm excited to give a talk on graph AI to enable precision medicine. I hope that those on Zoom can also hear me and see my slides, and if not, feel free to let us know. So throughout the talk, I will highlight various projects and papers and data sets, and all that materials and supporting resources are available to our lab website. So if you go to Zitnik Lab HMS Harvard EDU, you will find pointers as well as tutorials for those students in the audience who are interested in trying out some of the data set, working with some of the data sets or experimenting with methodology and algorithms that will be highlighted and discussed today. Okay, so with that, let's begin. Let's see if this works. Okay, great. So over the last few years, we have seen tremendous surge in interest of leveraging advanced analytic methods, and in particular machine learning techniques, to learn from biological and medical data. And so the kind of data and problems that are of specific interest to us relate to modeling complex systems that are pervasive in biology at multiple scales of organization. And in complex systems, we typically think of them as very rich network structured, graph structured data sets that can be used to represent cells, cellular interactions, proteins, interactions between them, and complex systems higher level of organization, to the level of individual, what kind of diseases might exist within an individual person, and all the way to the level of healthcare systems. And so those kinds of multimodal, heterogeneous, and interdependent data sets, when represented with graphs, yield interesting questions of how can we then leverage advances in machine learning and artificial intelligence to be able to learn and reason on those biomedical networks in order to produce relevant predictions, patterns, and insights. And so the emerging area of graph artificial intelligence, sometimes also referred as geometric deep learning or graph neural networks, is the area that has supported the development and use of machine learning on graph structured data. The use of geometric deep learning and graph neural networks have really skyrocketed over the last few years behind some of the most important advances and uses of AI in life sciences were graph neural networks. Here I'm showing just three concrete examples. The top one is showing AlphaFold. Part of the backbone of AlphaFold neural network is actually an equivariant graph neural network that takes as input a one-dimensional sequence of amino acids and produces as output three-dimensional map of coordinates of heavy atoms of a protein. An important structure in that neural network is a graph neural network. Second is molecular graph embeddings. Optimized to graph neural networks have been successfully used to prioritize molecular graphs in large chemical libraries, and that has led to the development of novel antibiotics. Halicin is the most important successful example of that. And thirdly, the use of transfer learning across different biological contexts defined by cell lines and or chemical and genetic perturbations have advanced understanding of cellular function, the study of pleiotropy in genes, and other cellular phenotypes. So what is the key idea of graph neural networks? So the key idea behind all these different applications is this notion of neural message passing or graph neural networks. So let me try to illustrate that through this one slide. So graph neural network is a nonlinear mapping function, here denoted as F, that takes as input a graph shown on the left. That graph represents a complex system of interest. Circles denote nodes or vertices, and edges indicate links. And graph neural networks will then map each and every node in that input graph to a d-dimensional vector, a vector of d numbers that is typically referred to as an embedding. And so the idea is that the way GNN model will do that, it will do that in such a way that two nodes in the input that have similar local network neighborhoods, they will be represented by points in the embedding space that are close together in that embedding space shown on the right part of the slide. So the way GNN is optimized is then such that the range latent embedding space is optimized such that it reflects topology of the input graph. So that once we are performing linear algebra operations with those embedding vectors for nodes, that reflects the structure of the input graph. So a variety of different kinds of architectures in this area, different specific algorithms have been developed to then adopt and work with different kinds of graphs and to encode different kinds of network similarity in the input graph. All those architectures are based on the idea of neural message passing. So what we will do is define neural messages that are these d-dimensional vectors that the neural network will propagate along edges of the input graph, and then we may transform them in such a way that the latent embedding space will be most useful for downstream applications. So the broad implications of this field of research is that with graph neural networks, we can now apply deep learning methods much more broadly than what was possible before. So traditional deep learning was primarily for analysis of images. So think of convolutional neural networks or transformer architectures, and analysis of sequence data through LSTMs and recurrent neural networks and other kinds of algorithms. With the GNNs, we can now use deep learning more broadly, and we can apply it to any interconnected graph structure data set. So this creates exciting opportunities in biology and medicine, and I would like in this talk to highlight a few of them and motivate the use of why graph AI is specifically relevant and a productive strategy to use for certain kinds of problems in biology and medicine. So why graph AI? What is particularly additional utility of those techniques? When we are thinking of modeling problems that require modeling the structure, geometry, symmetry, or prior biomedical knowledge that is naturally given. Okay, so just a moment, we need to figure out something.
That'll do it.
Sorry about that. The gain was maybe turned up too loud on the microphone input under the podium.
I see. Sorry to interrupt.
Does that work? Okay.
Yeah,
just give us a second or two.
Seems quite good now.
Great. Okay, great. Thank you so much for this technical help. Let's proceed. So we left with the discussion of what are the kind of problems where graph neural networks would excel and are particularly suitable to use. And so the kind of problems that they are particularly interested in and relevant to use are those that require us to consider the structure, geometry, symmetry, or prior knowledge of a problem that we care about. So in the context of modeling genome, it's when we are interested in modeling regulatory relationships or genomic dependencies, that's what we can effectively do with GNNs. Questions related to modeling molecular graphs or protein structure that require us modeling spatial distance or bonds are also naturally represented as graphs, as well as settings where there is an amount of prior biological knowledge or principles or rationales that we want to incorporate into the design of our machine learning model to make the model be more broadly generalizable. Our graph AI methods are excel. One of the second reason why we are excited about graph AI is because these models allow us to more readily translate predictions into actionable hypotheses. And I will return to that throughout this talk. The idea here is that what we care a lot about is not only producing models that achieve high measures of accuracy, but directly produce outputs that can lend themselves to actionable hypotheses in the laboratory. So that for us means that predictions must be interpretable, must be as specific as possible, and must be contextualized in the context in such a way that it is more readily clear what kind of downstream experiment could be done to validate hypotheses produced by the model. Another very important use of graph AI is in settings where we have large unlabeled data sets, or where our data sets are relatively small and therefore do not have sufficient amount of labeled data that would allow us to use standard supervised learning algorithms. So we have seen that many recent advances over the last decade in these areas have depended on methods being trained on large data sets with richly labeled inputs. So an example here is ImageNet, a data set with millions of images that are richly labeled that have then enabled the use of many deep learning strategies in the context of images. However, what I would argue is that many problems at frontier of biology and medicine, and some of the problems that we care most about, come with either very small data sets or data sets that are large but not highly accurately labeled or not labeled at all. And so in those contexts, graph AI can be really helpful. And so in this talk today, I want to broadly talk about two topics that we are working on in my research group. In the first part of the talk, I will show how graph AI can be helpful in assisting with diagnosis of rare genetic diseases. And in the second part of the talk, I will show how graph AI in graph neural networks in particular can help us predict therapeutic use of novel molecules as well as drugs that are already on the market and extend their therapeutic use for underserved patient communities. So the motivation for the first part of talk, which is that of assisting diagnosis, is the observation that one of the biggest challenges in medical diagnosis is being able to assist in diagnosing patients that are otherwise very hard, difficult cases. And so the particular disease area that we are interested in are rare diseases. There are over 7,000 rare diseases in the US, and they are defined as diseases where each affects less than 200,000 individuals. Most of these diseases are phenotypically very diverse and heterogeneous. And the challenge is that many frontline clinicians might not have prior experience with those individual diseases, and that can contribute to the difficulty in diagnosing patients, in recognizing their diagnosis in patients. And it often requires specialist, subspecialist, or multidisciplinary referral to diagnose the patient. And so the problem with that is that leads to diagnostic delay that can be so pervasive that it creates problems for patients. Patients might need to take redundant tests or undergo medical procedures that might not be necessary had we known their diagnosis early on. There might be substantial delay in providing appropriate therapeutic interventions or managing the disease. And sometimes the disease can progress in such a way that the window for optimal intervention has been missed because of the delay in diagnosis. And so this creates lots of questions whether AI techniques can be developed to help and assist with diagnosis of those patients. So those who might work in this area might be wondering, well, but there are already many, many methods that can assist with diagnosing, providing medical diagnosis. And that is true. AI has demonstrated tremendous success in assisting diagnosis. However, much of that has depended on the availability of large label data sets. One of most well-known example is a tool that has been in February of this year approved by FDA as the first autonomous AI tool for diagnosing diabetic retinopathy. And it's a neural network that was trained on over 110,000 labeled images of patients with diabetic retinopathy. And so that is great, but in settings where we have so few patients per a condition, it is unlikely or impossible to be able to train those models and use those models in the same way as it has been done for some of more common diseases. So we have developed an algorithm that's called Shepherd. Shepherd is a few-shot learning graph algorithm that can provide multifaceted diagnosis of patients with rare genetic diseases. It can be integrated at multiple levels and stages of diagnostic process of rare genetic diseases in the context of UTN. Let me give an intuition of how Shepherd works. So Shepherd is a graph neural network that is trained in 2 stages. Stage one. Stage one is to construct a large biomedical knowledge graph. So this is a multimodal knowledge graph of biological facts and known relationships between variants, their phenotypic effects, genes, and biological pathways. Take that knowledge graph that has no patient-level information. Using that prior knowledge alone, we pre-train a graph neural network in a self-supervised manner such that it can capture the relationships or those known biological facts. In the second stage, we do the following. We take the model from the first stage, and we use it as initialization, on top of which we can now consider patient-level information. And so for every patient, we represent a patient by a set of patient phenotypes that are HBO terms and a set of genes that are harboring variants found in that patient through genome sequencing. So then a patient becomes then given to those sets describing a patient becomes a subgraph of the knowledge graph activated by those nodes that represent phenotypes and genes harboring variants in that patient. Because we have prior embedding of knowledge graph, then we can really very easily produce embedding vectors for individual genes, phenotypes, and patients as a whole. And we can do that in such a way that when this second stage is completed, the model can be easily queried for novel patient, and then calculating proximity between embedding vectors in the embedding space, we can then nominate what are the diagnoses and what are possible causal genes that the patient might, that are relevant for the patient. The question that you might have had when I was describing the second stage is, well, but how can we train this patient embedder in step 2 if there are so few patients available for each and every disease? So here is the trick that Shepherd introduced. And so the main trick here was that we're actually training the model on a large number of simulated patients. So we rely on databases of rare disease information, such as Orphanet and Biobank and others, to simulate tens of thousands of patients that the model in step 2 is being optimized for. And so when that step is concluded, the model is fully trained and can be used directly to evaluate and make predictions for real-world patients. So in our case, we were collaborating with the UDN network, which is Undiagnosed Diseases Network, a nationwide initiative with 12 clinical sites throughout the US that is admitting patients that are hard to diagnose, and it is helping them provide and find the correct diagnosis through a lengthy and complex diagnostic process. And so we were wondering whether we can evaluate and take patients that were undiagnosed in UDN and run them through trained Shepherd model in an effort to provide medical diagnosis for those patients. And so we evaluated Shepherd on in 2 external cohorts, in addition to the UDN cohort of 465 patients in the UDN with diagnosis across 12 clinical sites. We also have another cohort, MyGene2, that the model was independently evaluated on. And so results of that evaluations have shown that this strategy of training graph neural network and bringing in prior knowledge can really help us train models much more with less labeled data. After having seen only a handful of patients with a given diagnosis, the model can correctly prioritize causal gene close to the top priority list. And so there were a few different tasks, 3 tasks in particular, that we evaluated Shepherd on. The first was causal gene discovery. And the task there for Shepherd was, given a patient represented by the set of their phenotypes and set of genes harboring variants in those patients, can we prioritize and produce a ranked list of those genes in an effort that at close to the top of the list would be causal gene for that patient? So what we found is that across 465 patients that were already diagnosed in UDN, for 75% of those patients, Shepherd was able to prioritize the correct gene among top 5 genes that the model has prioritized. And so this is really a good result because it shows that the correct relevant gene that was found very close to the top of the list. Importantly, those genes tend to be relatively far away and not directly associated to any of the phenotypes that patients have. That's not surprising because those are very hard, challenging cases to diagnose. And it was graphs that was a technique that enabled them to bring seemingly disconnected set of phenotypes and genes closer together. Second, what Shepherd can also do is to support patients like me queries. So in this way, you can think of Shepherd as a query engine that takes as input a patient and produces an embedding for that patient, take that embedding signature and use it as a query against embeddings of other patients in a database, and therefore find what are other patients that are similar to the query patients. And that is helpful because it can help inform diagnosis and downstream interventions. And finally, the third task is a novel disease characterization or characterization of diseases that have unusual representation, where Shepherd can be of value because it can generate these low-dimensional embedding spaces of both of genes, phenotypes, and patients, and therefore a query patient can be explained in the context of nearby phenotypic nodes or points in the embedding space. So key takeaway messages here are the following. So with Shepherd, we were able for the first time to bring deep learning to rare disease community. And that's really exciting to us because it shows it shows the promise of deep learning and most recent advances in machine learning for this area that has been underserved by prior efforts. The use of graphs is not only beneficial, but it's necessary to solve these diagnosis tasks. Shepherd overcomes limitations of traditional machine learning approaches in 3 ways. First, it can infuse prior knowledge of biology into a model through embedding a knowledge graph. It leverages self-supervised learning to align patient, gene, and phenotype embeddings. And it can be trained on a large cohort of simulated patients, meaning that we can answer new queries in a zero-shot learning manner. It can generalize to novel phenotypes, genes, and diseases. And more broadly, it demonstrates the potential of shortening the diagnostic odyssey for rare disease patients, which is an exciting opportunity for us. And we are working closely now with UDN to see to what extent we can provide Shepherd as a tool that can be routinely used by UDN researchers. So when the diagnosis of a patient is reached and identified, the obvious next question that emerges is whether there exist therapeutic interventions or what kind of novel therapeutic interventions can be developed to help reverse the disease effect or treat symptoms. And so in the second part of the talk, I want to illustrate how when we can use graph neural networks in a thoughtful way, we can advance the process of discovering safe and effective therapies for important areas. So we have recently started an initiative that serves as a meeting point between, on one end, AI machine learning scientists who are designing new algorithms, and on the other hand, biomedical and scientists and biochemists who are identifying meaningful tasks and problems in drug discovery and development And that effort, known as Therapeutics Data Commons, serves as a meeting point between two communities that have been otherwise quite separated from each other in an effort to facilitate both algorithmic innovation and advanced therapeutic science. So Therapeutics Data Commons serves as a AI foundation or platform for therapeutic science. You can learn more about it if you go to tdcommons.ai. And it comes with a very rich ecosystem of AI-ready data sets, ready-to-use Python packages with implementations of best-in-class algorithms for tasks, as well as leaderboards, benchmarks, and so on. The way we organize therapeutic data commons is by recognizing that many uses of machine learning in this area can be decomposed into a typical workflow. So a typical workflow in drug discovery would require the following steps. First would be for machine learning scientists in collaboration with biomedical partners to formulate meaningful machine learning task, a question that is at the cusp of benefiting from machine learning analysis. Those are questions that have sufficiently large and ready-to-use data sets that can be trained by the model. And those are questions also for which we are able to evaluate outputs, predictions produced by the model, and know how to go about evaluating them either quantitatively or experimentally. Once that is identified, one needs to source and process, harmonize the relevant data sets. Third, develop or implement machine learning model in any of the popular frameworks such as PyG or PyTorch, TensorFlow, JAX, Hugging Face. And once that models are developed, of course, evaluate performance of the model, understand its failures and successes, compare it to alternatives to decide what is the best-in-class algorithm that can be transitioned into practical implementation. So TDC supports development of novel ML theory and methods with a strong bent on understanding what are the specific algorithms that are most suitable for specific tasks and why would that be the case. So to do that, it provides support throughout all the stages of machine learning workflows, including meaningful mathematical definitions of relevant tasks, machine learning ready data sets that are harmonized, standardized, and can be easily linked to additional external databases that you care about, a variety of functions that can split the data sets into training, validation, test sets in a way that is close to real-world use of models, various methods for model performance evaluations, and various leaderboards where if you're interested in a specific task, you can go and see what is the currently best method for that task that the community has and use that method directly for your scientific question. Data sets in TDC cover a range of therapeutic modalities. Those that are currently supported include small molecules. Most of data sets cover small molecules. However, there are a handful of data sets that included in TDC and cover biologics, antibodies, and gene editing therapies. And those machine learning tasks, 22 of them that I've been mentioning, they go across stages of drug discovery, and they cover tasks related to target discovery, identification of candidate drug targets, to modeling activity and supporting high throughput screening, to predicting efficacy and safety of candidate chemical compounds and matching their therapeutic signatures with clinical outcomes, and tasks related to synthesis of therapeutics. It is an open science initiative, so those in the audience who might be interested in these questions, you are very much welcome to check out TD Commons and get engaged with us in a way that works best for you. What I want to mention in the next few minutes are some of the compelling use cases of the commons. And so what we have seen over the last year and a half is that the broad user base of therapeutics data commons have been using the resources that are provided by the commons for a variety of different tasks across stages of drug discovery and development. That includes modeling and predicting molecular properties in early stages of drug design, using models to prioritize therapeutic targets based on the strength on binding to relevant to relevant compounds, repurposing already available drugs for novel uses and to tackle and provide treatments for emerging pathogens, and be more broadly modeling and predicting indications and contraindications of drugs. Let me highlight some of those talks, some of those use cases. So molecular property prediction is a test that is concerned with predicting various endpoints for given only molecular structure of a small molecule compound. So typically that means that we're interested in building a model that will take us input a molecular graph, and it will predict endpoints. Most relevant and widely considered are ADMET endpoints that are concerned with properties around absorption, distribution, metabolism, excretion, and toxicity of that compound. And so the idea here is that if we would have such a highly accurate model for predicting those endpoints, we could apply that model across millions of molecules in chemical libraries with the purpose of finding those molecules that have acceptable ADMET profile, and those would be the molecules that would advance the therapeutic discovery process. So in TDC, we provide over 22 data sets that cover around 10 million chemical compounds across various ADMET endpoints. And they are all very easily accessible. And essentially with just 3 lines of code, one can get any of those data sets pre-processed and harmonized across those 5 types of endpoints. And so what TDC then provides are implementations of graph neural network models that take us input those molecular graph structure and predict these multidimensional ADMET endpoints for millions of molecules. Implementations of all of those methods are already available to TDC and can be readily used by users in a direct manner, or can be directly queried, or can they be further fine-tuned based considering your data sets beyond those that are in TDC. Benchmarking rigorously methods for these problems, we found that in fact, there is no single method or type of architecture that performs the best across all scenarios and endpoints. But the general principle is that pre-training is very helpful and boosts performance of the model. So taking large standard chemical libraries and pre-training the model to capture molecular graph structure is then very helpful and can help then take the pre-trained model and fine-tuning it on a small label data set. And that typically leads to better performance for the set of molecules of interest. And pre-trained graph models in particular produce strongest predictors overall. They outperform those expert curated methods that have been developed through decades of research in chemoinformatics. And they can do that because they were developed and inspired by the way those classic molecular fingerprints, such as Morgan or Smiles fingerprints, have been designed over decades of research in chemistry, and then graph neural networks provide the differentiable counterpart of those methods. Second, very widely relevant and use case of the commons relates to the problem of predicting interactions and bindings, in particular, between a candidate drug and a therapeutic target. And so the setup here is slightly different. The way you can think of it is that our input has 2 components. The first input is the structure of a candidate molecule that's given in the form of a graph where nodes represent atoms and just indicate bonds between them. And the second input is the structure of therapeutic target. And so both of those inputs are embedded into low-dimensional embedding vector by separate graph neural networks. Once those 2 low-dimensional embedding vectors are produced, a scoring function is then trained in such a way that the dot product between those 2 embedding vectors, the blue and yellow one in this slide, will be very high, where when the target and the compound are likely to bind, and will have low value otherwise. And so that score then reflects the binding affinity. So this setup is then supported in TDC, and a variety of GNN architectures can be used to embed molecular graphs or target proteins. The way we are evaluating those models is that we want to evaluate them in their performance in settings that are challenging and settings that are indicative of more realistic use. Or most practically, that means that we want to test models on structures that are very different than structures the model was trained on. And so in machine learning language, that means that we want our models to be robust, and we want our models to be robust to distribution shifts. So so those distribution shifts are the distribution of molecular structures the model has seen during training might be very different than the kind of structures the model will see during test time. And so we want to take our data sets and split them into train, validation, and test sets in such a way that molecules with similar scaffolds will be fully contained in either training set or in test set. So that in that way we can help benchmark performance of the model when it is asked to generalize to molecular structures that are very different than what has been seen during training time. So what is shown on this heat map on this slide is the following. So we have in rows, we have various methods that are designed for handling distribution shift problems. On the x-axis in columns, we have years, and years indicate the data set split. So for 2013, the first column indicates performance of the model in terms of binding affinity prediction. Higher value indicates better performance when the model was trained on molecules from that year and tested on molecules from the same year. And the further we go to the right part of that heat map, we see the performance of the models where the model was tested on data from between 2013 and 2018, but tested on data that was generated a year later, or 2 years later, or 3 years later. So here's what we see. So first we see that there is some variation in performance between rows. That's not surprising. Some methods perform better than other methods. In particular, ERM, or empirical risk minimization, is a strategy that works surprisingly well for handling distribution shift problem in binding affinity prediction. The second observation is the one that we can see that the performance drops drastically when we go from left to right within a row. And so what that indicates, in fact, we could summarize this number, we would see that out of distribution performance drops considerably on average from 43.6 to 33.9%. And so that essentially tells us that in many ways, when we are reading papers, writing them and reporting this AUC area under precision recall curves and other performance metrics that are near perfect, that does not mean that those models also perform so well when they're practically used. And we see how the performance degrades considerably when the model is asked to make predictions that are truly novel for very different kinds of inputs the model was trained on. So TDC supports that kind of analysis and provides practical insights into what are then specific algorithmic innovation one can make in this area. Finally, I would like to spend next few minutes highlighting one of the study that is relevant for later stages of drug discovery and development and after the drugs are already approved. And so that's the work that we have done in 2020 and expanded in 2021 as part of a COVID-19 task force to use and integrate, bring together a machine learning, automation, and novel data sources with the goal of providing support for rapid therapeutic innovation. So in many settings, traditional iterative development, experimental clinical testing, and approval of new drugs completely from scratch is not feasible. So the statistics is that it takes on average between 11 and 16 years and costs $1 to $2 billion to design a new drug completely from scratch and bring it to the market. So when that is not practical or feasible, which might be the case for certain therapeutic areas such as rare diseases or emerging pathogens, where we need to develop and deploy therapeutic invention as quickly as possible, an enticing alternative becomes that of drug repurposing. Can we take a drug that is already on the market, that was initially designed for a specific purpose, and extend its use? Figure out if any of those drugs could also be repurposed and used for in a different for different indication. And so that is the question that we're asking ourselves. And what we demonstrated in the study is that through careful use of ML and novel data sources and very close partnership with experimentalists, it's possible to cut down the time and resources needed for identifying drug repurposing opportunities considerably, to months or even weeks in certain cases. So let me tell you a bit more about this particular study. So the motivation was, or the big goal in question was for us was to find what are the compounds, chemical compounds that could be used to intervene against the COVID-19 infection. So our broad reasoning for that and the approach, the strategy that we were considering was the following. Since diseases disrupt normal behavior of genes, we want to find what would be then the drugs that could intervene against the disease by restoring the function of their disrupted genes. But what does that mean in the context of this virus? So in the context of the virus, that means that we were primarily working with the viral interactome and information on 330 human proteins that the virus attacks, and then the knowledge of how those human proteins interact with other human proteins in the cell by seeing where do they map when we overlay them on the human interactome or human protein-protein interaction network, which is a network where nodes are human proteins and edges would indicate physical interaction contacts between those proteins. And so we can study that viral interactome then in the context of known targets for each and every of approved drugs. And the hypothesis then is that the proximity between targets of known drugs and proteins that are attacked by the virus can tell us some meaningful information about the potential for that drug to be repurposed for the disease. And so this hypothesis is not new to our study. It's been founded and used by many other repurposing efforts, particularly in network medicine. So our approach was, considering the entire range of diseases and known approved treatments for those diseases, we want to prioritize compounds for this new pathogen, and we want to do that in a zero-shot manner. Zero-shot manner means that, well, we want to find then the compound for that novel pathogen, yet there are no examples of successful compounds that one could use as labels to train supervised models. And so we need to do that with zero labels or in zero-shot manner. So here's how we can do that. So the approach that we use is few-shot learning. This is a strategy that is that refers to a class of algorithms that are designed to work well when the amount of labeled examples is very small. We typically refer those labeled examples as shots. So few shots would mean you only have a few shot labeled examples for each class that you care about. So typical terminology in papers that you might have seen is when somebody refers that they have a 3-shot problem, that means that they are trying to classify objects into whether they belong to a class, yes or no, but they only have 3 labeled examples for each of those classes. So it's a hard problem. So few-shot learning allowed us to be able to identify drug repurposing opportunities for this emerging pathogen in the following manner. The process proceeds in 2 stages. The first stage is called meta-training stage. This is a stage where we intentionally expose machine learning model to the kind of really hard, challenging tasks that are similar to what the model will be asked to do at test time. So we design a number of challenges that are called training tasks, and each training task is centered around a different disease, a disease that has an approved treatments for. And so in training task 1, the model is asked to, after given only a handful, in this illustration here too, examples of drugs approved for disease 1, do the best it can to discriminate between drugs approved for disease 1 and those that are not approved for disease 1. You can imagine that the model would perform really poorly after training task 1. But the purpose is we will take the model and its weights at the end of training task 1 and use them to initialize model weights for training task 2 and repeat that process now in different disease. And then repeat that many, many times. We repeated that around 20,000 times. And when this process has been completed around 20,000 diseases, the model transitions into the second stage, which is the meta-testing phase. And at that phase, the model can be directly used and queried against the pathogen without having seen any labeled example or example of successful treatment, and produce a prioritized list of drugs for that pathogen. The method that can do that, it's called GMeta. It's an approach that's very flexible and scales very easily to very large graphs. We have a detailed description and characterization of the method in a NeurIPS paper that we published in the same year. I would highly encourage those who are interested to read it out as well as ask me questions about it. But we were able to show that theoretically and from mathematical standpoint, and provably that this algorithm can perform well. But not only that, we were then interested in actually using the algorithm for prioritizing compounds for COVID-19. So the first test that we have done is just to check whether the prioritized list of drugs that the method produced overlaps with those drugs that were considered as clinical trials at the time. And so that's a very, very weak way of evaluating the model, because the fact that there is a clinical trial on a compound does not mean that that compound would actually be safe and effective for patients. Still, we have seen some enrichment of top-ranked predictions shown here in this ROC plot on the left of GMeta models relative to more classic drug repurposing techniques that are based on network medicine, network proximity, and direct overlap between therapeutic targets and disease-associated genes. And so that has provided some confidence so that we were able to proceed to the next stage, which was to share those predicted lists of compounds with our collaborators. So our collaborators in the National Emerging Infectious Disease Laboratories in Boston then took top of the ranked list made by the model, and those were 918 compounds, and then they screened those compounds for efficacy against the pathogen in African monkey cells, followed by experiments in human cells. So the results of that were the following. By screening top-ranked predictions in human cells, we obtained near 62% success rate. And so success rate was defined as the success was defined as the rate at which the pathogen its growth was inhibited when the drug under different dosage levels was administered to the cells. In contrast, if that screening process would not be guided by priority lists produced by our GMeta algorithm, the hit rate or success rate one would get would be around 0.8%. So this is an order of magnitude higher hit rate among top 100 drugs than alternative approach. Importantly, what we were interested in knowing is there something common to those drugs that the model predicted and that were experimentally verified? Are there some common biological mechanism or some common pattern that the model has relied on when making those predictions? And so we used GNN explainers and explainability techniques that we've been developing in the group to probe graph neural networks, distill their predictions, and figure out what parts, regions of the input graph the model is focusing on when making predictions. We found that 76 out of 77 predicted drugs that reduced infection were such that their therapeutic targets did not coincide at all with those proteins attacked by the pathogen. Instead, there was a series of cascading effects between targets of those drugs and then those indirect effects that eventually reached proteins attacked by virus. And so that plus various additional analysis that we have done that I can that we kind of thoroughly describe in the paper. I'm happy to ask questions about them. Have motivated us to realize that that those drugs rely on network-based action. They could not be identified by more classic molecular docking strategies, and we refer to them as network drugs. We have since then extended this approach more broadly beyond COVID-19 to other disease areas, and particularly to rare diseases, where we are modeling then predicting therapeutic use of already available drugs in the context of perturbation experiments, where we take response signatures of chemical perturbations and overlay those gene signatures on top of biological knowledge graph, and then measure proximity between signatures on the knowledge graph to prioritize drugs. So the results of that approach are particularly promising for those diseases that have very few annotated treatments, or for those diseases that are for which the knowledge describing them is relatively sparse, meaning they are found in parts of the knowledge graphs that are very sparsely connected to each other. And in particularly because graph neural networks can take into account those indirect effects, not only direct effects that are available about a specific molecule, the performance of the model is particularly strong for those diseases that have few treatments or limited molecular understanding. So here on the right, I am showing evaluation of the graph neural network in 3 specific scenarios. Performance is measured as area under precision recall curve, so higher value indicates better performance. And we can see that few-shot learning performs comparably well and performs actually on par with standard supervised learning methods when the model is asked to make predictions around diseases with many annotated treatments and considerable prior biological knowledge. But the gap between performance of standard approaches and few-shot learning approaches increases drastically when we go and use those methods for harder settings. And in various evaluations we've been doing, we have seen these effects across different disease areas. We have seen those effects for when the model was asked to predict indications, so positive use of a drug towards treating a disease, as well as contraindications. And we have since then been engaged in a big larger effort on evaluating predictions made by the model across disease areas against EHR data sets at Mount Sinai Hospital in New York City. And practically that means that our users of our models are clinical researchers who interact with our models through visual interfaces. An example is shown here on the slide, and they can ask questions in natural language. For example, will clozapine treat unipolar depression? What could the disease treatment mechanism would be? And what the model would provide as output are prediction scores, visualization of embedding spaces, and visualization of parts of the knowledge graph that the model has relied on when making the prediction. And so that has been really helpful for our users to decide when to trust predictions or not trust predictions. And we've done recently a user-centric study with our collaborators, Niels Gellenborg lab in my department, showing that presenting the outputs of AI analysis to users who might have limited machine learning experience in that way can help them identify what are those predictions that are most trustworthy that are made by the model. Okay, so with that, let me wrap up today's presentation. My goal was to give an intuition behind the use of machine learning for complex systems and networks and graphs in particular. I hope I convinced you that graph neural networks and more broadly neural message passing and graph representation learning techniques can be helpful in areas of precision medicine with an illustration of facilitating diagnosis for rare genetic diseases and areas of therapeutic science through the various use cases of therapeutics data commons. The papers, codes, data sets mentioned today are all available on our lab website. Also go to tdcommons.ai therapeutics data commons website. We have a separate initiative called AI for Science Initiative, which is an initiative of 27 academic groups around 4 continents, and we are carefully asking ourselves what are current uses of AI that are successful, and more importantly, are there examples or problems where the use of AI can be counterproductive, harmful, or not beneficial, and what are the challenges and concerns there. And more about that effort is also available at AI Science for Science community GitHub AI.io. My lab is hiring, so if you are looking for postdoc positions or there are outstanding PhD students who would like to work with us, reach out. We have several open postdoctoral fellowships in AI for cancer drug discovery and immunotherapy analysis, as well as we're recruiting PhD students every year. Thank you.
So any questions? And people on Zoom, you can just ask question directly. We can hear you really well.
Hello. So I think it's pretty fascinating to see the GNN application on the part 2 of your talk about the therapeutic, and you has mentioned that the GNN can help to just find like the drug target for the emergent like pathogens, and I'm just wondering like how well like the result of the prediction can resist the mutant of the pathogens or the variants of the disease?
Great question. That's a question that we are asking ourselves as well. So we have an ongoing project where we are studying mechanisms of resistance with a particular focus on TB, leveraging some of the expertise in TB in the department at Harvard, where I am. And initial results are promising in the sense of that we can, once the large data sets are available, we can identify these models can predict accurately resistance, finding patient samples whose response to certain antibiotic treatments will result in antibiotic resistance. That said, however, we don't have a good understanding of those mechanisms for the emerging pathogens project that I was highlighted today. That was completely independent.
So yeah, great talk. I guess my question is, so on slide 25, when you were talking, I think it's slide 25, when you had that big chart with all of those predictions, I guess my question is, so you explained that the drop in performance is because of, it's in distribution from 2013 to 2018, and then out of distribution from 2019 to 21. But why is it getting worse between 2019 and 2021? Because it's like 2021 is far worse than anything else, at least as far as I can tell on that chart. So like, why is it still getting worse? Like, wouldn't we be improving
A great question. So so your interpretation is correct. The worst performance would be is achieved by methods in last column. So you can see that the average Pearson correlation coefficient, which is measuring the agreement between predicted binding affinity scores and ground truth binding affinity scores. So the lower value indicate worse performance. We can see that for the last column performance of models, no matter what models you pick, is really poor. So why does the performance go down as years go up? So performance going down because the model is asked to predict further in the future. And it's asked to do so in such a way that the test molecules for the 2021 year have different scaffold structure than those molecules that the model was trained on from year 2013 to 2018. And so the shifts in the underlying data distributions of the structure of those molecules, think of the distribution of occurrence of certain functional groups or motifs in those molecular graphs changes drastically. And because many of these algorithms are still relying on those that signal to make predictions, they are performing increasingly poorly where they are asked to generalize further in the future. And further for the future for us is a proxy for the test molecules having more different structure than those molecules the model was trained on. So you could think of this slide as showing negative results. So the current state-of-the-art methods perform in such a way that when you expose them to challenging settings that are those that drug designers would care about, they perform really poorly. So like this project for us was particularly motivated by this observation that when you are reading various papers for binding affinity prediction, you see those papers reporting near perfect accuracy. And so the question is, why are then those models not more broadly used in drug discovery? Why have they not transitioned into practical implementation? And so what we realized doing detailed analysis of predictions is those models make really good predictions for inputs that are trivial. Those kinds of settings where a test molecule is very similar to a molecule that's in the training set plus minus 1 or 2 bonds. But those are trivial predictions because drug designer, biochemist who works in that therapeutic area is most likely to already think of that structure by themselves. They would not need an AI model for that. The use cases where they would benefit the most from AI models would be to quickly sweep through areas of the chemical space that's not densely investigated by existing experimental data sets, and in that areas find promising or non-promising chemicals. And what we see is that state-of-the-art models do not perform well in those cases, which to us, it provides some insight into why we don't see their broader adoption. And more concretely, we are now working with the drug designers on improving some of these methods.
So I have a question on the Shepherd model. So you mentioned that because this is targeting for rare disease, that's why you have rare samples as inputs to your training GNN, and we use like simulated patients to this model. So will we have problem of overfitting? If so, how to avoid this?
Great question. Short answer is yes, this can be a problem of overfitting. So here's what we have done to address this problem, and particularly in our evaluation studies, we did 2 kinds of experiments. So first is that the knowledge graph and simulated patients that were generated were generated using the data available up to June 2021. And the test set of patients that the model was evaluated on was that of patients that were admitted to the UDN and diagnosed later. So that is one way how we were trying to mitigate the potential data leakage. So the kind of data leakage that one could happen is, imagine a patient admitted that has not yet been diagnosed, it seemingly has a diagnosis that's very hard to discern, and they are admitted to UDN. However, parts of the insights and in terms of the relationship, the phenotypic effects of their variants might have been characterized already, and that data would be then uploaded to particularly OrphaNet, which is this repository for rare disease research. If that information then fits into our generation of synthetic patients and training of the models, that would lead to data leakage or overfitting, as you referred to it. So the way we mitigated that is by doing this temporal split of what the data used to train the model was generated up to a certain time point, and the model was evaluated on patients later that were tested admitted later on. Second, we have also used UDN on a cohort of patients that are currently undiagnosed, and we are following up on predictions for a small number of patients for them with experimentalists. And third, in addition to UDN, we have second independent cohort of MyGene2 patients, which is a patient community portal where patients are volunteering their own data in an effort to be diagnosed and biomedical community provide support for them. And so that's a separate effort, a separate cohort that we evaluate our model as well. So yeah, we are very much aware of this problem, and we have lots of checks in the paper that to mitigate those effects.
So I have a question for you, Gil.
Yeah.
I'm interested to know if these graph neural networks are related to what around here is called hypergraphs. Been used quite extensively by one of our colleagues, Ndika Rajapaksa.
Yes, so the question was whether these graph neural networks are related to hypergraphs, and the answer is yes. So hypergraphs are higher order graphs that can model not only pairwise relationships, but relationship between triplets of nodes or quadruplets or higher order interactions, and are particularly useful for problems such as modeling polypharmacy or drug combinations, multigenic interactions, and so on. So graph neural networks are a class of algorithms that can also work on hypergraphs, which are a specific type of graphs. Actually, they have been recently a series of I think very interesting papers where graph neural networks trained on hypergraphs were used to model higher order chromatin interactions. That was done primarily by Jian Ma from Carnegie Mellon University, and his group has published a series of papers demonstrating the use of graph neural networks trained on those higher order graph structures, and they were able to successfully predict chromatin interactions and do also multi-way learning with chromatin data and histone modifications.
So would they
Thank you
add
for
any value to what you've been doing?
Yeah, for us, hypergraphs have been is something that we were considering and have projects on for modeling drug combinations. I didn't talk about that effort today, but an important direction in our group is to support with AI post-marketing surveillance of drugs. And so for us, that means that we're working with data from FDA National Adverse Event Reporting System, which is large observational data sets that we are trying to deconfound them in different ways to identify if there are patient groups that for which there are certain adverse events overrepresented or underrepresented when they're taking multiple drugs at the same time. And so we are representing those interactions between drugs for those patients as hypergraphs of drugs that the patient is currently taking on, and then predicting labels for them, which are adverse event outcomes.
Sounds very good. Thank you.
Okay, there's one more question from Zoom. Joey, can you ask the question?
Okay, yeah. Hi, Professor Zitnik. Thanks for the great talk. So I have a question regarding the distribution shift part. So I'm wondering, how did you divide this training environment for this environment risk minimization model?
Yeah, so the question was of how do we split the data set into training, validation, and test set for our distribution shift study. And so for this study of predicting binding affinity under distribution shifts, the data set was split based on molecular scaffolds. So that most practically means that one can use chemoinformatics tools such as RDKit to compare for a given pair of compounds their molecular backbone. And so cluster compounds bear those backbones. And so then you take an entire cluster, which is a cluster of compounds with similar backbone or similar scaffold, and you put the entire cluster either fully in training set or fully in test set. And so in doing so, what you end up producing is a training, validation, and test set where molecules with similar scaffolds are all used only for training or only for testing. So that's one type of split, which is called molecular scaffold split, that is implemented in therapeutics data commons. So you can readily use it, just write through a few lines of code on your own data set that a distribution shift was evaluated on. We have a few other kinds of data set splits. And another one was temporal data split, where we have obtained information on when certain patents were filed and finalized for therapeutic targets, and then we were splitting the data based on the years in which those patents were granted. So that's another type of split that we were considering as well. It's also implemented in therapeutics data commons.
Yeah, thank you.
One more question. This will be the last question.
Hi, excellent presentation. I did have several questions, but I'll stick to one for here, and maybe if you still have some time later, I might want to ask a few more. But I'm just wondering, for the second part of your talk, where you talked about applying the models to, repurpose therapeutics or discover new therapeutics for treating diseases, I was just wondering, have you tried to incorporate data on maybe natural product compounds or, uncharacterized novel compounds? Because I guess I'm trying to understand what is the baseline information that you need for a single compound for a model to work effectively. How well does it perform with maybe compounds that are not as highly characterized as, already approved therapeutics?
Yeah, great question. So in short, a couple of points. So first is in the work so far, we have not been considering natural compounds. We have a bigger collaboration with partners where we're looking actually at food ingredients and that can be decomposed into chemical compound signatures and then thought of as some form of therapeutic intervention in the right dosages. So, but that's a bit kind of unrelated to the topic of today's presentation. The second point that I'd like to make is that one of the things that we are very excited in the context of TDC is that we can stratify those machine learning tests along different stages of drug discovery. And so that means that we are then being thoughtful in the sense of, if we are designing a model that is helpful in stage 1 of drug discovery, that model could only use the data that is readily available at that stage. We're seeing many of the pitfalls of published literature in this area be, for example, modeling molecular property prediction tasks using data on toxicity that's only available when the molecule enters the animal testing mode. And of course, that kind of use of the model, or like developing a model to solve a task in stage 1 using the data that's only available in stage 3, the drug discovery, it's really not particularly meaningful thing to do. So one of the efforts that we are then undertaking is to systematically evaluate the utility of different data types for specific machine learning tasks. That can be helpful, and also all those insights are available to Therapeutics Data Commons website. What we have found so far that some of the users of the commons have used that information to decide given limited resources available, what kind of experimental screen might be most informative to perform for a specific group of compounds that they are considering.
Okay, since we're almost 20 minutes late, so let's stop here. Let's thank Marinka again for this wonderful talk.
Thank you.
Very, very good.